Hello everyone, welcome to today's live broadcast, using genomics to understand human health and disease. I'm Rachel Brown of LabRoots, and I'll be your moderator for today's event. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Dr. Richard Myers. Dr. Myers received his BS in biochemistry from the University of Alabama in 1977, his PhD in biochemistry from the University of California, Berkeley in 1982, and he did his postdoctoral training at Harvard University. He joined faculty of the UCSF Medical Center in 1986, moved to Stanford University in 1993, where he served as professor and chair in the Department of Genetics and director of the Stanford Human Genome Center. Since 2008, Dr. Myers has been president, director, and faculty investigator of the Hudson Alpha Institute for Biotechnology, a nonprofit research and teaching enterprise in Huntsville, Alabama. I will now turn it over to Dr. Myers for his presentation.
Okay, thank you, Rachel. Good morning, everyone. I'm going to tell you about how we use genomics and genetics to study human biology problems, both health and disease and basic biology. But I'm going to give you a little background on who we are first, just because many of you may not have heard of Hudson Alpha Institute. It's, as Rachel said, it's a nonprofit research institute focused on genomics and genetics. We're funded by grants and philanthropy, sort of a soft money place, foundations and some support from the state. We have 10 faculty members in our midst now, but soon about 15 or maybe 16. And we, because we do genomics and large-scale problems, both in human biology as well as in plants and other organisms, it's a very collaborative field, so we work with people from all over the world. An unusual thing about Hudson Alpha is that we have biotech companies as our associates. They're tenants on our same campus. They're in the same building, same campus. There are 28 of these, and they all are involved in life sciences problems. Here are the faculty. Some of you may know some of these folks, and we just now recruited a new group of 5 faculty members from Wisconsin that will be joining us this summer. Here's our campus. I'm always proud to show this. It's this flagship building here is where, on the lower left, is where most of the research goes on. The second building behind that has some of the biotech companies, as well as our data housing, a conference center as well. And we always like to point out this is the world's largest double helix. It's a walkway that's a mile and a third down and back. That's sort of the center of our park. One thing about the way that we do our work here is, and many others I think that do large-scale genomics, is we've centralized all of our wet lab research, especially in ultra-high-throughput sequencing. This is run by Dr. Sean Levy, who is one of our faculty members here, a very talented individual with a very modest-sized team that runs now 24 next-generation DNA sequencing machines, including the new Illumina X10, which is the highest, fastest one. We've done lots of functional genomics, as well as lots of whole genomes and partial genomes. I'm going to tell you a little bit about that. That's a photo of their team. And all of our work that we do in-house, that we initiate, and collaborators are done through the services lab to make it centralized, and centralized data, primary data analysis, as well as even some of the detailed analyses. But I should also say that this team here. It's a services lab, does many, many collaborations as well as fee for service for literally hundreds of, now more than 2,000 projects and people from all over the world. All right, so I'm going to tell you about, to talk about sort of two areas that I sort of artificially divide, genetics and genomics, and they're obviously very related. But let me talk about genetic analysis. And geneticists have been essentially genome scientists for many decades, long before the Human Genome Project. But what's happened is that because of increased knowledge as well as vastly increased technology, we can do this on a much larger scale now. So when I think about genetics, I'm thinking about differences in DNA sequence. That's allelic variation. So sequence variants, and of course, most of us think about those that we're born with, we got from each parent, one or either parent or both parents, as well as somatically acquired ones, especially in cancer, but the immune system undergoes somatic mutation as well. And there are probably others that we don't even know about yet. And what's happened in recent years is that we have for at least a couple decades, a little longer, have looked for genes and identified genes involved in at least in humans by having candidate genes and doing linkage analysis, et cetera. But this has changed now because we can look at targeted regions and the whole exome, all the exons, encoding regions of the genome, and now in our hands with the new approaches that we're doing, whole genome sequences are frankly to the same cost, if not even less expensive than whole exome. And then there are other ways to think about this, because sometimes we're looking for not just single base allelic variation, but bigger changes, copy number variants and other rearrangements. And sequencing can identify those, but there are other tools that we use and the field uses to really work on those. So let me tell you about one example of targeted sequencing. We do this a lot for one gene or sets of genes and sometimes hundreds of genes. But this is a very interesting one, and it's technology that many others are around the world are developing and using. One of the, I think, early proposers and adopters of this is Jen Han, one of our faculty members, a very talented entrepreneurial and creative scientist. And he basically developed methods for amplifying specific segments of the genome by multiplex amplification methods, and started applying this to a problem that people have been really interested in for a long time, which is identifying, can we take peripheral blood from an individual, human, that can be other organisms too, and determine all the antibodies and T-cell receptors, at least key components of our immune system. Can we identify all of those? And the concern years ago was that this may be impossible because there are 10 to the 15th or more possibilities with all the rearrangements that can occur in both of these gene systems, as well as in antibodies, the hypermutability that occurs, and the other kinds of changes that occur in T-cell receptors. And so with ultra high throughput sequencing, it became possible to start thinking about doing this. So Jen developed ways of taking a small amount of peripheral of blood, looking in the periphery and saying which antibodies and T-cell receptors are there. So the interesting thing, and he's been applying this really for 10 years, but especially these last few years, is sequencing has become cheaper and cheaper, and we really can do this. So we now routinely, Jen's group as well as others in many collaborations, take a little bit of blood and in two separate types of reactions, measure, amplify, and then sequence about 5 to 10 million sequence reads for the T-cell receptors and for antibodies. And this lower part of this slide shows the panel up there just showing the top, let's say 500 or so T-cell receptors, and the height of the bars are how much of each of those are made. And what Jen noticed very early on when he started looking at different diseases is that when essentially every disease looked at so far, you get a greatly reduced repertoire. Now, so look in this lower panel, lower part with colon cancer, you see just a few clones that are expressed at higher levels. These other clones that are present in normal tissue are also present there. They're just present at a lower fraction, considering now that these newer ones are being made. And the sort of hypothesis is that when you have something that goes wrong, that you start making, your immune system starts to try to deal with that by making antibodies sometimes, or T-cell receptors, or both. And you see this reduced repertoire. So here's another way to depict this. In this next slide, it shows 4 individuals, and each one of these little oval colored figures is one of those T-cell receptors, in this case, T-cell beta receptors. And if you look at each of these, this is maybe the top 500, or I don't know how many, about 1,000. You see that there's an enormous amount of diversity. You could sequence deeper or make a bigger picture and show many, many more that are present at lower quantities. But these are the sort of the top few hundred. And you see a lot of diversity. But if you look at a disease, and this one, and certainly if you look at cancer, you look at many other diseases, you see a greatly reduced repertoire like I showed in that earlier slide. And so you see large clones appearing that are essentially look like they're overtaking for the 5 million or whatever number of sequence reads that you're doing. These are the ones that end up dominating. And again, you see that everywhere in every disease. And just another view of one of the individuals who does not have a disease, and showing the difference in the diversity. So here's the thing that surprised me when Jen started doing this, and we've started to apply this to others as well, to dozens and maybe 100 or 150 individuals with particular diseases, started seeing some specific T-cell receptors and antibodies that show up in different individuals. So this top panel here of maybe 10 individuals, 12 individuals, you see in breast cancer, you see some T-cell receptors, and you can't see it from here, but if you look at the sequences of those women share similar amino acid sequences in their T-cell receptors. Same is true for antibodies as well. So that was interesting, because the idea that you might be able to find disease-specific signatures in the bloodstream within cells, actually. So you're not looking for cell-free DNA or RNA. You're actually measuring it out of RNA from the cells. So that, to me, makes it a very, very valuable tool. And so we're applying this for all sorts of phenotypes, especially diseases, but pre and post drug treatment, following the progression of disease, whether something is recurring. And we think it's going to be one of the most powerful tools, as many other groups are doing as well. I will say one thing about these, the amino acids, you heard me say the amino acid sequences of these variable regions are identical or very similar in different women with breast cancer, let's say in this case. But what's interesting is that the nucleic acid sequences are different because of the redundancy of the genetic code. You can get the same amino acid sequence. To me, that speaks very strongly that this is biologically relevant. That's convergent evolution. You're converging in on T-cell receptors or antibodies the same way to try to attack a problem in different individuals. So now I'm going to tell you about more now genome-wide sequencing, and it's to solve or to work on a problem that has been around for forever, but now is tractable. So here's just a little bit of information. This is a childhood genetic disorders project that's led by Dr. Greg Cooper, who is one of our faculty members here. I and Greg Barsh and many others here participated in this project. I'll tell you more about the people in a minute. But here's the problem, is that if you look worldwide, about 2 to 3% of kids who are born, live born, often will have a problem, sometimes serious, sometimes milder. And these are ones that are noticed at birth or not long after birth. Many of those have to do with the nervous system. Our neurons are probably the most delicate organs in our cells in our bodies. And so often you have intellectual disability, intellectual delay, developmental delay, other things. These are often syndromic. You often have other things that you can see in the kids. The guess is, and now the data are proving that the guess was that most of these have a genetic contributor at least, and maybe even a genetic cause, meaning a single gene or a small number of genes. This is important because, for one thing, while 1.5%, 2%, 3% doesn't sound like a large number, maybe in the aggregate, that is a huge problem public health problem worldwide. And there are many things that happen with this. So you don't know the cause of most of these. I forgot to mention. We know the causes of maybe 5 or 10% of all of these disorders. The vast majority, we don't know the causes. We don't know the genes that are involved, if there are genes. But the problems are that when parents have a child like this, they often are at their wits' ends about trying to figure out what's going on. The doctors can't diagnose them. It's not a mistake that the doctors are making. These are very heterogeneous, maybe thousands of different causes, and they don't have names on them. They have general, often similar phenotypes or similar symptoms. So what happens is the parents don't know, the doctors don't know. They go from one doctor to the other to the other. They spend years on doing lots of testing. Sometimes these are invasive tests. You can't make any kind of prediction if you don't know the cause about how this is going to progress or what's going to happen. Certainly don't have much that you can do for treatment other than trying to treat the symptoms. We don't know anything about how to treat, how to think about new therapies if we don't know the causes. Parents are trying to decide, do I want to have more children or not, given that we've had this problem? And of course, there's a serious problem of parents feeling that it's their fault. So if you can figure out the causes of these, it's extremely helpful, even if you can't help the person, the child, or the individual with their clinical outcome. At least it helps in some of these problems. And as I'm going to tell you, we've actually are able to accomplish both. In some cases, we actually do have impact on the clinical on the treatment outcomes. So we started this project less than 2 years ago. We're calling it the Hudson Alpha Pediatric Genetics Project. It's to recruit 450 children who have these disorders that are not Down syndrome, Rett syndrome, Fragile X syndrome, other ones for which we know the causes. These are the unexplained ones that do not have a diagnosis. We're also recruiting both parents, and consented both parents. And one of the things that we're doing, and it's our first cases of doing this, where we're also now both doing research on how to deliver the information back to parents and physicians and patients, as well as actually doing that, returning the information. So this is a group of from Dr. Martina Bevan leads the clinic here in the Huntsville, North Alabama Children's Specialist. She has more than 1,800 patients in her clinic, showing you that this is a widespread problem. We're also working with Dr. Kyle Brothers at University of Louisville, who is a bioethicist as well as a physician, and he's studying how do we best return this information to maximize helping the families and the patients and the physicians. And then our group here, as I mentioned, led by Greg Cooper, where we do the sequencing, data analysis, and interpretation. So this slide may be a little confusing. We're about 200 trios into that sequencing, both the children and both the children and both parents. We're finished sequencing a couple hundred of those, and I've only fully analyzed about 120 of them, showing you pointing out what one of the big issues here is. The sequencing now, it's not that it's trivial, but it's quite straightforward. We have, with Sean Levy's team, have a very, very high quality, very rapid sequencing, and even primary analysis of calling all the variants. But then figuring out which ones of these might be the cause or contributor to the child's phenotypes, that's the hard part, this interpretation part, and that's where we spend much of our time and money. But if you look at these numbers here, actually on the left it says no variants. That's not quite true. What it means is that, and I should say, these first 121 families are just exomes. We started out doing exome sequencing. Once we bought in December into the Illumina X10 system, where we can sequence whole genomes cheaper than we can the exomes, we started sequencing those. So we haven't analyzed the whole genomes or interpreted them yet, or they're not shown in these slides. Of course, these individuals, these 57% individuals, do have variants. We all have sequence variants, but there were none that were suggested to be pathogenic. How you determine whether something is pathogenic or not is a complicated and important problem that our whole field is working on. Greg Cooper is an expert, working with colleagues in Seattle, Jay Shinduri in particular, an expert in helping to figure out those kinds of, does this sequence variant, this base pair change, is that likely to cause harm to the gene, not to the individual, to the gene? So that's one of the big problems. Liz Worthy, one of our new faculty members who is moving here in about a month, is also an expert in doing that kind of analysis and interpretation. But even if you take the weaknesses that we have there now and look at them, almost 40% of them, of the individuals that we sequence, we're getting either a definite or likely diagnosis. We're figuring out a gene that is the cause or contributor to their phenotype. So 17% are absolutely, we know are pathogenic and feel very confident. 8% are almost 100% confident. And this VUS stands for variant of unknown significance. These are variants that clearly harm a gene, but we don't know yet whether they're ones that actually cause the phenotype. If you think about this, these are rare disorders, sometimes very rare disorders, and the loss of function mutations that we're finding in these genes may have never been seen before. But as the databases and throughout the world get populated, and as long as people share data, which we hope they will, we'll start finding these occurring more in more than one individual. And the sort of thought here is that occurring in 3 individuals with a similar phenotype is enough to make the call that it is indeed the pathogenic cause of the disorder. It's a little harder for dominant mutations, but that's part of the problem as well. So this is exciting because we're not the only ones doing this, obviously, but our group is one of the big groups, and the group joining us from Wisconsin are very big in doing this. So we're doing this for research, making discoveries. Every one of these genes new genes that is found is a biological problem to study as well. And I will say, well, let me just show you a couple of these. Here are a couple of examples where Greg and his team found the actual cause. And so one of them was a girl who had mild intellectual delay, but was not diagnosed with Rett syndrome, which is a relatively rare, but a usually easily diagnosed syndrome. The physicians seeing this girl are very good at this. They know Rett syndrome. They know how to diagnose these disorders. But this girl had atypical symptoms. And yet, when we sequenced her, she had a mutation in MECP2, which is known to be causing that particular disorder. It was a milder mutation, and that knowledge allowed the physicians and the family to provide their daughter with some treatment options that had not been thought of before. Here's another example where we had 2 unrelated individuals here in Huntsville who had mild intellectual delay as well, no seizures, but when their genes were sequenced, when their genome was sequenced, or their exome, mutations were found, and clearly they had mutations in a gene that in others are known to cause seizures, in fact, usually really severe seizures. So these children were then analyzed by EEG, and sure enough, were just subclinical for having seizures, and are now being treated to help prevent the seizures from ever having an onset and the consequent possible brain damage that comes from that. There now, this data, our data have grown, and Howard Jacob and his group from Wisconsin coming here have twice as much of those data, and in multiple cases now we're finding examples where knowing the answer now has an impact on the clinical outcome, and in a few cases incredibly dramatic impacts, saving lives. It's not that it happens every time, but it does happen enough to make this absolutely both rewarding and worthwhile. One of the things, because we're sequencing both parents, is that we get what we call incidental findings, and that means mutations in genes. And this is a little controversial. What is actionable? And you can debate this forever, but if you have a mutation in a BRCA1 or 2 gene and you're female, you might want to at least know it, and you might want to do something about it, preventative or otherwise, or certainly increased surveillance. Lots of places where, and there are many genes where if you have a mutation in them, usually loss of function ones, that you would have a cardiovascular problem, and you might want to watch after it. We've had 2 cases where people had mutations in the ryanodine receptor gene, which if you have a mutation, loss of function in that gene, it encodes a calcium channel. You get something called malignant hyperthermia. If you get particular types of anesthesia, which are actually commonly used, your temperature goes up to 112 degrees and often die on the operating table. So 2 of these adults we found unrelated and different mutations in their genes are now wearing bracelets, or at least have warned their physicians that they are at risk for this disorder. All right, now I'm going to tell you about how we've applied in a big collaboration genomics to identify causes or at least contributors to diseases. So this is Lou Gehrig's disease, or ALS. Many of you know about it. It's actually about 5,000 or 6,000 cases in the U.S. Diagnosed every year. People live with this disease for 2 to 3 to 4 years usually. And it's constantly recurring mutations happening. So we started a project with Biogen IDEC in Cambridge, Massachusetts, led by Dr. Tim Harris. He's runs their genetics program there, and then collaborating with David Goldstein and his group when they were at Duke University. David and many of his people are now at Columbia, and then our group here at Hudson Alpha. I will say this was led by Liz Cerulli, scientist in David's group, and Brittany Lassane, a postdoc in my group, and they working closely together with each other, with the Genome Services Lab here and Sean Levy, and Tim Harris's team did this large project that we just recently published. It's not in press, it's actually out. We sequenced about 3,000 individuals with ALS. This was exomes. We certainly do whole genomes now because it would be cheaper, and then had more than 6,000 controls. We got some findings based on those by doing a burden analysis. We identified genes that were likely contributors to the disorder. We followed up with custom capture sequencing of additional cases and controls, and then analyzed them for the disorder, obviously disease burden, but for things like age of onset and gender, et cetera. And what happened on this next slide, you can see it was a little fuzzy, I apologize. You see in this QQ plot, you see when you get off the diagonal here, those are potential genes, or those are contributors. And so we identified a dozen or more that are genes that are contributors to this disorder, sometimes even out-and-out causes. In the upper right corner there, you may not be able to read it, but that's SOD1. That's actually the first known cause or gene to be identified years ago as being the cause of this disorder. So it was gratifying that we got the known gene because we didn't screen the patients out for those, and we got them at the expected frequencies. So we published this recently in Science, one of the novel genes, and it was highlighted in the papers, TBK1, and it's a protein that interacts with optineurin and these other genes, two genes that are known to be associated with ALS, and these genes and it play a role in autophagy and inflammation, which I guess is involved in almost in many disease phenotypes. But a lot is known about this, and there's some functional analysis of this in model systems that has corroborated that this is clearly a new target. And the idea behind, and the reason Biogen was and is interested, they work on neurodegenerative disorders. The reason they're interested in this kind of project, and they funded this as an academic style project, and the data are published and released and available, which I think is quite noble of them. But the reason they're interested is that they're looking for new targets. These disorders are very hard to manage, and anything that helps to give an idea of how you might do therapy is extremely helpful, and especially when they tie into a pathway where multiple members of the pathway are involved. And truthfully, some of these could be useful for other kinds of diseases that have similar phenotypes, not just necessarily ALS. Okay, I'm going to now talk about gene expression. So now, instead of genetics, I'm talking about how I kind of artificially break this into genomics. This is functional genomics. And I think we all have known for a long time that you could look at messenger RNAs, for example, and you can see differences between different individuals, different tissues, and certainly the same tissue, disease and non-disease. So clearly this variation in expression is important, and it contributes to phenotypes that we care about. So I love to show this because it's the extent of my talents in artwork is that I did 20-something years ago, that we know that the eukaryotic genome has genes that are split and have introns often. They have a start site of transcription, sometimes multiple start sites. And right at the beginning of the gene, of course, is where RNA polymerase and many components bind to then begin transcription. We know upstream from the gene, within the gene itself, and downstream from the gene, there are little segments of sequence that bind proteins that also regulate gene expression and regulate other DNA processes. If you look at what we call the cis components, these are the parts of the DNA itself. We know we have about, from the Human Genome Project and much work beyond that, we have about 21,000 protein coding genes. We probably identified very close to all of those. There may be some that code for small proteins. There are thousands of, and we don't know the real number of this, of non-coding RNA genes. And unfortunately, at least for the scientists, that the regulatory elements, these little non-coding elements that are places where DNA proteins bind, there are probably a million or more of those. They're spread all throughout the genome, and every gene may have multiple of these, or does have multiple of these little segments that bind proteins. The proteins are the transacting components. We know that there are at least 1,600, probably more, genes that code for proteins that bind to DNA. Most of those are probably involved in transcription, obviously others involved in DNA replication and recombination and repair and other processes. Most of these recognize little cis-acting elements, maybe 6 base pairs, 8 base pairs, so tiny little segments that are these sequence-specific DNA binding proteins bind to. And then some of those, of course, histones and the various versions of histones, RNA polymerases, others, are general. They bind to lots of places, not necessarily in a sequence-specific manner. So if you have any doubt that transcription is important in human disease or other diseases and other organisms too, this is an old example, a gene found by Len Pinocchio when he was a graduate student in my lab in the mid-90s, that mutations in this gene lead to a form of epilepsy in children that's progressive, called progressive myoclonus epilepsy. He found the gene on chromosome 21. It's a small gene, and when you look at mutations, this is a recessive disorder, so you have a mutation from mom and from dad. When you look at mutations, there's stop codons, splice-like mutations, etc. But the majority of the cases are actually a promoter mutation that occurs in almost 90% of the cases. Every human Mendelian disorder that's been found, with 1 or 2 exceptions, have promoter, enhancer, and other regulatory mutations. So clearly this is just as important, if not more so, than the exons themselves. That was maybe a trite point, because I think everybody recognizes that, but it really is an issue, because we don't know where all those 1 million segments are, and it's hard to figure out how they all work together. So I refer to this as global genomic analysis, or functional genomics, where we're looking at the readout of the genome. And of course, that's messenger RNAs, all the other non-coding RNAs, like microRNAs, small ones and big ones as well. DNA methylation, chromatin variants, and all the different kinds of epigenetic things that you can have, chromatin accessibility, and then proteins that bind to DNA, each of these transcription factors and general factors. Can we look at these globally? And of course we can now. These are based on sequencing methods like RNA-seq, methyl-seq, DNA-seq, many other methods have been developed. And then what you do is you do these experiments to look genome-wide at these events and then try to figure out what they mean. And that's actually, frankly, the hard part, because we're having to deal with lots of complicated data that's not bimodal, the way an allele is either there or not there, whereas all of these are quantitative events that occur sometimes in cell type specific fashion. That makes all of this complicated. Nevertheless, we and others in the ENCODE project and others as well are trying to attack this the best we can. So let me give a few examples of looking at this. So transcripts are important, clearly. Here's a case where K.T. Varley, who was a postdoc in my lab, now a professor at University of Utah, discovered in breast cancer, a big collaboration we have with physician scientists at University of Alabama at Birmingham, discovered fusion transcripts in breast cancer. 7 transcripts that occur in breast cancer that are breast cancer specific are semi-specific. And they occur in cases, this is not looking at rearranged chromosomes like the Philadelphia chromosome where the DNA is changed in cancer, but where you have 2 genes facing in the same direction, relatively short distance between those, a few kb at most probably, where you get a transcript starting in the first gene, and instead of cleaving and doing poly A addition at the end of this, it continues transcription and it ends up splicing into the adjacent gene. So we call these transcript fusions. We published this recently, and here's some of the information. So 3 of those we focused on because they were particularly interesting. These 3 in particular showed a stronger specificity towards breast cancer as opposed to other tissues. And the reason this is potentially interesting for these 3 is that all 3 of them make fusion proteins where one of the partners is a transmembrane protein. So the hypothesis or the idea is that you're making a new protein that's not seen in normal cells, and outside of the cell is a peptide where the fusion occurs that is not normally seen in normal cells. And so that's important and potentially interesting because the idea that you might be able to use this as a therapeutic. In cancer, at least one of the hopes of being able to treat and prevent maybe cancer is to do something where you attack the cancer cells with antibodies in particular, specifically. And so the idea here is to make an antibody or a set of antibodies that recognize those aberrant proteins, and because they're outside the cell, it's a much easier thought of how you might deliver it. The antibody binds, it's taken up into the cell, you have a toxin on the antibody that's then released and kills the cancer cell. So we're collaborating with the pharmaceutical company to try to see if this will actually work. So I'm going to tell you about the protein-DNA interactions. This is something near and dear to me. I want to be sure I get through the talk here, so I'll speed through. But ChIP-seq is a method. Barbara Wald's group and my group were one of the first. There are several others who 7 or 8 years ago developed this as a way of really trying to look genome-wide at where where a particular protein is bound to the DNA. So we call these interactome measurements or genome-wide occupancy measurements. And this is just looking at the short arm of chromosome 6. You're seeing little peaks. There's a lot of computation and analysis and controls that go into getting these data. But as you look down on the lower right and then the lower left, a higher and higher resolution, you can actually get one of these little hills here is one place where that particular transcription factor, I guess there are 5 or 6 of them here, where each of those factors are bound. So you can get this snapshot of where a protein that you're interested in interacts. So we're doing this as part of the ENCODE project. It's only one of the many things that ENCODE is doing. We've been part of this group for a long time. This is an encyclopedia of DNA elements, and this is similar to the Human Genome Project, a project where it's a consortium, it's a group of scientists generate large amounts of data for DNA protein interactions, for RNA, for DNA hypersensitivity, et cetera. All of these types of events, does them in a standardized way with the right kinds of controls, et cetera, and then releases that data for absolute free, immediate use by anybody in the community. And we try to learn as much of it as we can ourselves, but thousands of users are using this information, and it really is a community sort of a project where we're providing, this is meant to be a service to provide this to the community, and it's having a huge impact. Many of these regions are ones that are involved in disease, for instance, but also for basic biology, people or many people are using them. So that's where we're doing a lot of our ChIP-seq, and it's the hardest part about ChIP-seq is finding a good antibody. We've, between the groups that work at ENCODE and then even taking the entire rest of the world that is doing this, we're probably representing only about 1/4 of all the transcription factors, maybe a little bit more than that now. So we're really working hard to try to find more of these, and it's been diminishing returns because it's hard to get good antibodies. Some of these may not even be immunogenic enough to do it. So we started tagging the transcription factors in cell lines in recent months. This is being done with Eric Mendenhall at University of Alabama in Huntsville here, and our ENCODE group, Barbara Wald's group, others, etc. And so you add a tag to the gene, and by using CRISPR, you're tagging the endogenous gene, so you get normal levels of expression, etc. We've started doing this, and we've done it for modest numbers so far, about 20 so far, but are trying to turn this into a pipeline to do this for hundreds, if not many hundreds of them. And the thing that's gratifying, at least so far when we've done this with protein, with transcription factors that we know that we do have good antibodies for, we compare the antibody version, which we're calling wild type here, versus the flag or the tag version. And they look almost identical. So we think that this is a robust and a very, very good way to do this. So we're applying it to a bunch of those for which we had never gotten antibodies. I will brag on Dan Savick, postdoc in my laboratory, very talented postdoc who's experimentally amazing, but also works and analyzes lots of data. And he figured out a way. We were trying to do ChIP-seq in tissues, lots of different tissues, human and mouse, and could not get any of the one or two published methods to be robust or reproducible enough. And so he developed a method where it's very simple. You just have to keep everything cold. You have to put the tissues in a very non-destructible bag, that particular one is made by Covaris, and then hammer it with a hammer multiple times to pulverize the tissues. This now has led to lots of measurements in many different tissues by our group and others that are reproducible. Very, very powerful method, even though it seems so simple. Okay, a couple more points, and then I will end. DNA methylation, we measure this genome-wide by a variety of methods. And Brittany Lassane and Jim Brooks. Jim Brooks is a former colleague at Stanford University, a urologist and researcher, working on kidney cancer and prostate cancer. So Jim isolated 135 tumors from individuals who had renal cell carcinoma, and he had also matched non-tumor kidney tissues from each of those individuals. So Brittany measured DNA methylation in all of those samples, genome-wide, and then analyzed the data and compared normal, which this has gotten shifted out of whack here, I'm sorry, but normal versus the tumors themselves, and identified DNA methylation markers, about 20 of them, that absolutely distinguish a tumor from non-tumor. So if you look at this next plot on the left here, we have called an ROC curve. If you don't know what these mean, when you're looking at whether a test is going to be valid or not, you want the area under this curve to be as close to 1 as possible. And the red one, which is overlapping with the blue one, but the red one is her data that gave an ROC curve of 0.99, which is almost unheard of to get one that good. So her 20 markers were absolutely capable of distinguishing this is renal cell carcinoma versus not, and even for all different subtypes of renal cell carcinoma, there's several that are known. And then a few months ago, TCGA, the Cancer Genome Atlas Project, published data, or released their data. It wasn't a publication, released their data, and Brittany was able to do the same thing and look at it, and it very, very highly replicated the results. So we believe these are real. So what? Is it we knew these were tumors, so does it do any good to do that kind of measurement? Well, if you already know somebody has a tumor, it actually would be helpful in a biopsy or after the tumor is removed to actually learn something about it from looking at these markers, because they do distinguish some of the subtypes from each other, some of the markers do, and some of them actually have predictive power for whether the tumor is going to recur or not, which would affect a treatment. But the real reason this would be valuable is if we could find these methylated DNAs in the urine or blood from breakdown of the cells. We're furiously trying to do that. Others are as well. We're trying to apply that in that manner, because then you could imagine that being a screening tool to see whether someone has kidney cancer or not. This is true for kidney, pancreatic, ovarian, many other cancers where certainly early detection of any cancer would be helpful, but those three that I mentioned are particularly bad in that you often get big tumors already having spread by the time you get symptoms. So this is helpful. So, all right. Okay, the last thing I want to tell you about is using genomics, genomic kinds of measurements, and we do this mostly with RNA-seq and with methyl-seq kind of methods to see whether you can predict whether someone's going to respond to a drug or not. The pharmacogenetics and genomics question that many are interested in. I think this is potentially one of the most powerful approaches in genomics, and I'm just going to show two cases here where, again, this was done by Katie Varley when she was in the lab working with these physician scientists in Birmingham. And she basically found in one of their drug trials that they did it at UAB, they found it was an antibody drug to the death cell receptor. And they had women, they were in this trial for breast cancer, women were treated with this, and you had women who responded, they were sensitive to it, and they were resistant to it. And you don't have to really understand very much to just see that those two groups had very different methylation patterns. So some of these genes are actually interesting from the breast cancer point of view, and even potentially new targets. But that alone is a potential diagnostic or a tool that could be used to determine whether you'll have a responder. An even better one was one where at UAB the trial was done with a combination of two drugs that had not been used before, again with breast cancer, and this is ER-positive breast cancer. And KT found very strong signatures for women who responded versus those who didn't respond, and some of the responders were actually super responders. So this was strong enough to warrant a larger clinical trial. Clinical trials are expensive. This was done on a small number of individuals, expanding the trial. And where I think most of us see this going is that you get you use the test itself, the genomic signatures, like the ones we show here, to choose the people who will be in the trial because they're likely to be responders, and that increases your chance that the trial will work. Okay, so I've given you kind of an overview of the kinds of things that we in our institute and our collaborators in our labs here do, and mostly in human health and disease. This last slide just shows I do very little of this work. I help to coordinate and interact with lots of collaborators, and it's great fun doing this kind of work. Thanks for your attention. I'm happy to answer questions.
Great, thank you so much for that informative presentation, Dr. Myers. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is from Thomas from Planet Connect. Could promoter regions become demethylated to change from being silent to being oncogenic?
Yes, so certainly you get methylation changes happening in normal human biology and especially in cancer. So that absolutely happens where you see changes, and often it is in promoters, but it can be in other regulatory regions that are distant from the gene, where they get either hypermethylated, and usually in promoters, when you methylate, that means the gene is shut down. And so that certainly occurs in cancer and other sort of normal events like in aging as well as other diseases.
Okay, so it looks like that was the only submitted question, so we'll go ahead and wrap up. Thank you, Dr. Myers, again for that presentation. Today's webcast will be available for on-demand viewing through November 2016. You will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.